joined the lab a little bit earlier. So I'm really dedicated so yeah, especially for that part, you probably would. For like the coding.
All right, hello everyone.
You need to have taken like concept.
Welcome to our afternoon session. Thank you for attending our Heart Month kickoff day. Again, this is the second lecture of 4 lectures and some other community events that will continue next week at the Graham Center, so please join us for that. But this afternoon, I am delighted to present Dr. Brian Aguado, who joins us as an assistant professor of bioengineering at UC San Diego, where he utilizes novel biomaterial technologies to study sex-related differences in cardiovascular disease. Prior to UC San Diego, he completed his bachelor's degree in biomechanical engineering at Stanford, and went on to do his master's and PhD at Northwestern University, and then his postdoctoral research with Dr. Christiane said at the University of Colorado Boulder. Though he's only been an assistant professor for a couple of years now, he's already received numerous awards, including the Burroughs Wellcome Fund Postdoctoral Enrichment Award, the NIH K99R00, the Pathway to Independence Award, a Career Development Award from the AHA, and the Chan Zuckerberg Initiative Science Diversity Leadership Award. He's passionate about increasing representation and inclusivity in science. And as part of his efforts, he co-founded the Latinx Indian group, which is where we first met him a few years ago now, where the Latinx Indian group sponsored a symposium that was held virtually through FIU BME. He was named one of the 100 most inspiring Latinx scientists in America by Cell Press, and he received the Biomaterials Diversity Award from the Biomaterials Journal. So already a very impressive career, and I'm happy to have him today to share his research.
All right, everyone, thank you so much for coming today. It's such a delight to be here in person at FIU. Like Josh mentioned, I first interacted with this department to host the Latinx Indian Symposium, which I'll talk about a little bit later. But I'm super excited to also share a little bit about my research on understanding sex differences in cardiovascular disease, specifically aortic valve stenosis. And I will try my best to read our title slides. So we obviously have some projection sheets, but we'll kind of work through it. So the title of this slide is called Current Medicine, so one size fits all, right? So currently, most treatments for disease rely on the one size fits all approach. So given a population of patients, say with cancer or high blood pressure, more often than not, the patients are administered the same treatments. Sometimes those treatments can be quite beneficial for patients with the reduction of symptoms over time, whereas at times there might not be any beneficial recovery of symptoms, or there might be detrimental side effects. So why don't these medications work for everybody? So that's the title of the slide. So a recent study claims that drugs might not do what they think they do. So this recent study claims that the mechanisms of actions for certain drugs in clinical development are actually incorrect. So logically improving our understanding of how drugs work might logically improve our ability to target patients more effectively. Dosing is often based on the weight of the patient and rarely considers other variables such as sex and ancestry that are known to impact drug metabolism rates. Lifestyle factors, including high-fat diets, can also impact metabolism, so think about that next time you have your next Cuban sandwich, right? And asking too much of patients with complex dosing regimens. So this is especially true of the elderly population that takes multiple medications throughout the week, and at variable times throughout the day, so that might cause variability in how patients might be responding to their drugs. So that brings me to the concept of precision medicine. So I think that precision medicine considers individual differences in people's genes, sex, age, ancestry, environments, and lifestyles to make more effective treatments for individual patients. So the future of medicine is going to rely on these precision treatments, right? So I think that acknowledging patient-specific differences, we can design patient-specific therapeutics to result in a relief of symptoms for multiple different patients, irrespective of their sex, age, or ancestry, right? And I think that as bioengineers, I think we have this unique ability to enable precision medicine. And where we can start to think about designing different types of biomaterials-based systems to, as potentially diagnostics or therapeutics, to consider patient-specific heterogeneities. So I like to term these precision biomaterials. So the idea is that we can potentially customize biomaterials in such a way to evaluate patient-specific biology, or enables patient-specific treatments, or treatments based on subsets of patients, like groups based So that brings me to my formal definition for precision biomaterials. I think that these are tools that can enable our understanding, detection, or treatment of diseases in a specific patient or subsets of patients. So precision biomaterials lie on the spectrum. So to illustrate this idea further, we recently published a perspective piece that kind of defines this field. So we like to think of designing different biomaterial systems to address patient-specific differences, such as groups of patients stratified by sex, age, or ancestry, all the way to individual patients. And we like to think about tuning a number of different properties of our material systems, including material chemistry, including, for example, very simple materials, simple polymer materials to release drugs, all the way to more complex designs where we can potentially look at patient-specific enzymes to cleave off drugs at specific rates for that patient. We can also think about different fabrication strategies for our materials, so we can think of very simple scaffold materials all the way to three-dimensional adaptable materials that can grow with the patient, especially important for pediatric applications, for example. We can think of different bioactive components, including allogeneic transplantations. I'm going to move this, honestly, it's projecting now. It's just getting in the way of my laser pointer. So we can think about allogeneic stem cell transplantations, so from different patients, all the way to engineered cell transplantations from that same patient. And we can also think about potentially analyzing data from patients. So we can obtain patient biopsies from individual patients and analyze them on biomaterial systems, all the way to thinking about designing implantable material systems to provide real-time data for a variety of different biometrics, for example. So my lab, my fantastic team at UC San Diego, we use, so the title of this slide is using precision biomaterials as tools to dissect mechanisms underlying sex differences in cardiovascular disease. So we all know that cardiovascular disease is the leading cause of death in both men and women, yet our mechanistic knowledge of the sex-specific mechanisms that guide cardiovascular disease progression, particularly in women, remain poorly characterized simply because of our reliance on male bias models. So my lab in precision biomaterials, we operate at multiple different length scales to understand how sex hormones, sex chromosomes, the sex of the cells themselves and how they respond to drugs, inflammatory factors found in serum, and the global inflammation response in the patient, all cause sex dimorphisms in cardiovascular disease. And we use biomaterial systems to probe these sex differences. So today I'm going to share with you two vignettes that I think where we use biomaterial systems to try to understand patient-specific and sex-specific aortic valve disease mechanisms. So I'll start off by kind of sharing with you how we've used precision biomaterials to study sex-specific disease progression in aortic valve stenosis. So our lab is focused on aortic valve disease and how sex impacts aortic valve stenosis. So to get everyone on the same page, this is Miami Heart Month, so I'm sure everyone knows what aortic valve stenosis is. But just in case, so my lab focuses on the aortic valve that controls blood flow from the left ventricle to the aorta. And as this disease progresses, valve leaflets become diseased with age, among other risk factors, such as sex, ancestry, hypertension, and high-fat diets. And these valve leaflets become stiff over time and calcify. And due to this calcification process, patients can experience symptoms such as angioedema and severe heart failure. And this disease affects around 13% of the population aged over 75 and can lead to a very short mortality rate. And can also lead to other symptoms such as cardiac hypertrophy and fibrosis. And currently the only treatments available are surgical valve replacements or transcatheter aortic valve replacements. But not every patient is eligible for these procedures, and there might be opportunities for restenosis. So it's not the perfect solution for a lot of patients. So that brings me to the clinical problem for this study. There's still no general consensus on the drug therapy to slow or halt AVS progression. And to try to discover drugs or other small molecules that might inhibit this disease from progressing, we have to understand the cellular and molecular mechanisms by which it occurs. So the title of this slide is Fibroblast Regulate Valve Remodeling and Fibrosis. In the valve, we see we have this population of fibroblast-like cells known as valvular interstitial cells. And these cells, over time, and due to routine valve injury, they activate to myofibroblasts. And these myofibroblasts are known to remodel the matrix inside of the valve and maintain healthy valve homeostasis. But as these cells activate to myofibroblasts to stay chronically activated, these myofibroblasts contribute to the aberrant stiffening of the aortic valve and eventual fibrosis and calcification of the valve tissue. And you can see these sections from healthy and diseased valve tissue, where the diseased valve tissue expresses a high abundance of alpha smooth muscle actin, which is a marker of myofibroblast activation. And multiple microenvironmental cues can govern this VIC to act myofibroblast activation process, including a number of different mechanical cues, such as matricellular signaling, external forces due to fluid flow, due to hypertension, for example, and matrix modulus can all exacerbate the myofibroblast phenotype. And we can also think about different inflammatory factors that might contribute to this process. So to start to develop some model systems to try to understand the biological mechanisms, we need to engineer the proper tools. So if we were to take cells, these valvular interstitial cells, and culture them on tissue culture plastic, you can see that these cells just automatically activate to a myofibroblast. And this is likely due to the supraphysiologic stiffness of plastic. The last time I checked, our valves are not made of plastic. So that supraphysiological stiffness automatically activates these cells. So to solve this issue, my lab, in addition to a number of other labs, use PEG hydrogel systems. So these are water-swollen polymer networks where we can incorporate cell adhesive ligands to this PEG hydrogel backbone and culture cells directly on our hydrogel. And we can tune the polymer concentration of the hydrogel to increase the stiffness of the gel and modulate the stiffness of the gel to recapitulate the stiffness of aortic valve tissue. And by designing and engineering our gels, we can see that if we were to culture cells on the soft matrix that reflects the stiffness of healthy valve tissue, we can see that we can maintain fibroblast quiescence. But when we stiffen our matrix, you can see that the cells activate to myofibroblasts. So this gives us a really nice platform by which we can control fibroblast phenotypes and investigate the processes by which cells activate and deactivate. But this particular study, published in 2013, did not consider sex as a biological variable. And I'm going to show you how important sex as a biological variable is in aortic valve disease. So there's been a number of studies now that have demonstrated that there are clear sex differences in how valve disease manifests between men and women. We see that in this particular study, with age-matched and disease severity-matched patients, men tend to develop more calcified bone-like phenotypes in valve tissue, relative to women that develop more fibrosis, scar-like phenotypes in valve tissue. And this is shown in hundreds of different patients from this recent study, suggesting that men with, from young and old patients with bicuspid aortic valve disease, in addition to tricuspid aortic valves, we see this increase in calcification density in valve tissues. And Kristen Master's group at the University of Wisconsin actually was the first group to actually obtain male and female valvular interstitial cells and culture them separately. And she cultured them separately in tissue culture plastic and found a number of sex differences in gene expression signatures from these male and female valvular interstitial cells, suggesting that male and female cells are fundamentally different cells and should be treated as such. So it's becoming increasingly important that cell sex matters. So for example, we know that male and female cells can behave differently, and this is by and large due to sex chromosome biology. So we can start to think about how sex chromosomes, namely the X and Y chromosomes, express genes and epigenetic modifiers that might influence somatic cells' gene expression and cell signaling. And I'll come to show you that sex-specific intracellular mechanisms might influence how cells respond to drugs and might contribute to sexually dimorphic disease progression. So the title of this slide is Genes That Escape X Chromosome Activation Partially Drive Sex Differentiation. So for example, one cellular process that's unique in female cells is known as X chromosome activation. So I'm sure everyone is familiar with cute calico cats. And a really beautiful example in high school biology is the regulation of the fur color on calico cats. So female calico cats have this particular cat has a gene for orange fur on one X chromosome and the gene of black fur for the other X chromosome. So you can see this cat expresses splotches of orange and black fur. As it turns out in humans, there are a number of different genes that escape X chromosome inactivation. So this inactivation process leads to hundreds of different genes where there can be variable or complete escape, indicating that female cells can receive 2 copies of certain transcripts from the X chromosome, and they escape this process of X chromosome activation. And this process is common among a number of different tissues inside of female tissues. So it just goes to show that that genes that escape X chromosome activation can govern a wide array of sex differences across multiple different tissues. So after kind of exploring this literature, I started to think about the question as to whether or not cells from male and female valve tissues have these intrinsic sex-specific responses to our engineered hydrogels. So I came up with this hypothesis that increased expressions of genes that escape X chromosome inactivation might increase myofibroblast activation in female valvular interstitial cells due to that increased fibrosis that's observed in women, right? So the idea here would be that we can sex separate male and female fibroblasts and evaluate them in our in vitro models to understand how genes that escape X chromosome inactivation might govern this increase in myofibroblast activity observed in female cells relative to male cells. So we started to understand how, we started to evaluate and understand sex-specific differences in myofibroblast activity in our tissues. So we obtained tissues from both porcine and human sources and sex-separated them and stained them for alpha-smooth muscle actin. And our stain here is green for alpha-smooth muscle actin and blue are the cell nuclei. And we routinely observed increased expression of alpha-smooth muscle actin in all 3 layers of the valve tissues, the fibrosis, spongiosa, and ventricularis. And we observed increases in alpha-smooth muscle actin abundance in human tissues as well. And these are the quantified results here. And just for the sake of, for this presentation, the small dots indicate technical replicants, whereas the large dots indicate biological replicants. So, so that should hopefully kind of detail the level of data that you're able to collect in this study. So when we take these cells out of the tissue microenvironment and culture them on our biomaterial substrates that are intended to mimic the valve tissue extracellular matrix, we find that these cells maintain the sex differences that we're observing in tissues. Namely that when we culture cells on female cells on softer stiff hydrogels, we see routinely increased activation relative to males cultured on the exact same substrate. And we provided tissue culture plastic controls where alpha-smooth muscle actin, there was no clear sex difference observed in alpha-smooth muscle actin expression on TCPS. So these are the quantified results here where we have on the y-axis percent activation, or the number of cells that contain these beautiful stress fibers. And we see that female cells routinely have this increased myofibroblast activation on our hydrogel substrates. So after observing that phenotype, we next performed a transcriptomics analysis to try to understand the sex-specific transcriptomes that might be regulating this process. So we observed that in quiescent fibroblasts versus these activated vital fibroblasts, we saw a number of different genes that were sex-specific, in addition to a number of genes that were common between both sexes. And we performed an ingenuity pathway analysis and found, and the asterisk here indicates sex-specific pathways that were upregulated in female cells. But we were also curious about this pathway, particularly in the male data set. So in the male data set, we found Rho A signaling pathway to be one of the most enriched pathways in the male data sets. And we figured that, and Rho A signaling, Rho ROCK signaling is a common pathway that regulates myofibroblast activation. So we sought to validate this pathway experimentally. So this is a beautiful experiment that my undergrad, so there's a story behind this, behind Brandon. So Brandon was my honors thesis student at Coulter. And to start my research lab at UCSD, I basically packed him in my suitcase and brought him to San Diego, and he started the lab with me. So we've been working together for a number of years now, and so he started off as my staff research associate, and he's now one of my PhD students. So it's been terrific working with him over the past few years. But we conducted this experiment together where we started to evaluate how male VICs are actually more responsive to Rho A inhibition relative to female VICs. So the experiment was dosing in this Rho ROCK inhibitor on 2 male and female cells cultured on our tissue culture plastic or our stiff hydrogel. And routinely, we observed that male cells were more sensitive to the Rho ROCK inhibitor relative to female cells at higher concentrations on our tissue culture plastic controls. But however, when we cultured the cells on our stiff hydrogels, once again, these stiff hydrogels that recapitulate the fibrotic microenvironment, we found that male cells were more sensitive to the drug at lower concentrations relative to female cells that might seem to be unresponsive to this drug concentration, indicating that not only is it really important to separate our cells, but it's also super important to consider the microenvironment that we're culturing them in to generate more accurate in vitro models of how cells might respond to drugs. So why are female myofibroblasts less responsive to this inhibitor? So we next used our sequencing data to compare the genes that are upregulated in myofibroblasts, and we compared them to sex-specific genes. So we took our library of genes that were up and down regulated from female myofibroblasts and compared them to all known genes that escape X chromosome inactivation in humans. And we found a number of different genes that overlap between these two data sets. And just as a control to understand how Y-linked genes might influence myofibroblast activation, we didn't really see the Y chromosome to be involved in these processes. So in our female data sets, we became interested in two specific genes. One gene is known as BMX. So BMX it codes for the Rho tyrosine kinase receptor, and which has been previously shown to be necessary for angiotensin-induced cardiac hypertrophy. It would be a nice gene to potentially target to understand how myofibroblast processes are regulated uniquely in female cells. We also looked at the STS gene. So STS codes for steroid sulfatase, which is an enzyme involved in metabolizing steroids and plays this pivotal role in regulating estrogen and androgens. And recent work has also shown that STS overexpression can lead to increased expression of integrin beta-1s, which are super critical for regulating myofibroblast activation. So after identifying these genes of interest, we next tested the effects of knocking down the gene activity using small molecule drugs. So in this experiment, we tested the effects of ibrutinib, a drug known to target the BMX gene, and we tested irusostat, another drug known to target the STS gene. And when we dosed in the drug in our in vitro cultures, we saw a sex-specific effect. We saw specifically that female cells seem to be more responsive to this inhibitor in reducing myofibroblast activation relative to male cells, indicating that these two drugs might be regulating, they might be able to target this specific gene and reduce myofibroblast activation. So now with these two drugs in hand, we next did a combinatorial study to try to see if these drugs in combination would help reduce Rho-ROCK signaling as well. So we performed an experiment where we took our female cells, we also have male cells data, and I'll explain that in a moment. But in our female data set, we cultured female cells either using a vehicle control or our Rho-ROCK inhibitor at a constant concentration. And then we dosed in either ibrutinib or irusostat to knock down gene expression from the X chromosome. And we found that by doing this combinatorial drug treatment, we found that we could reduce female activation uniquely using these two drugs. In male cells, I'm not showing the data here, but in male cells, we didn't see this effect. So this indicates that this is a uniquely female mechanism. This is a unique mechanism to female cells. And to probe this mechanism further, we performed siRNA knockdowns of these specific genes. So this is qPCR data from male and female cells cultured on the hydrogels. And we find that by taking our control siRNA, we find that female cells do, in fact, express higher levels of BMX relative to male cells. And we can knock down the BMX gene expression in both the cells, and both male and female cells. But then when we look at the ACTA2 gene, the gene that codes for alpha small, we see that female cells once again have increased expression of this gene. But when we knock down the BMX gene, we see that we can see this unique reduction in alpha small active gene expression, indicating a sex-specific mechanism. We also performed the same experiment targeting the STS gene. So we also looked at siRNA and was able, by siRNA targeting the STS gene, and we were able to knock down both male and female STS expression, but uniquely reducing ACTA2 expression in our female cultures. So to kind of conclude this part of the talk, I wanted to try to introduce this kind of the mechanistic pathways that we're proposing. So in female cells with two X chromosomes, we have this double dose of BMX and STS expression. And we think that this increased expression of genes that escape X chromosome inactivation partially contribute to regulating Rho-ROCK signaling as the cell is experiencing different mechanical cues or biochemical cues. I'm not showing data from our endothelial or PI1 study, but we also found site-specific effects from these two inflammatory factors. Then all of these factors participate in Rho-ROCK signaling to regulate alpha-synuclein expression uniquely in female cells, relative to male cells that only receive one dose, and even if we knock these genes out, they don't participate in regulating alpha-synuclein expression. So with that, and that kind of set the stage for, so that was essentially all work that I did in my postdoc lab, and when I was like wrapping up my postdoc, I stumbled upon all these like sex differences, and was like, I can 100% start a lab in this area, right? So my undergrad, Brandon, who is now my PhD student, started to investigate sex-specific responses to inflammatory serum factors from aortic valve stenosis patients. So this is I'm super excited to be sharing this work because it's our first publication from our lab, and this is all Brandon's effort, which is I'm super proud of to share this work today. So aside from mechanical cues, inflammation is also known to be a hallmark of aortic valve stenosis. So during routine valve injury, we have these recruited monocytes that are recruited to the valve tissue and differentiate to pro-inflammatory macrophages. And this is a collaboration that we did with one of my close friends, Joe Grimm, where we tested the effects of different inflammatory factors secreted from pro-inflammatory macrophages and found the mechanism by which we think these myofibroblasts lead to osteoblast-like differentiation. And essentially, factors like TNF-alpha, IL-beta, or IL-6 regulate this activation from the myofibroblast all the way to potentially an osteoblast-like phenotype in these cells. So to study the role of how inflammatory factors regulate this Vic to myofibroblast to osteoblast-like differentiation process, we collected serum samples from aortic valve stenosis patients in collaboration with cardiologists at the CUH Medical School and also at UCSD to characterize the proteome of the inflammatory factors found in patient serum. And so this was in collaboration specifically with Professor Tim McKinsey at the CUH Medical School, where we've been collecting serum samples from aortic valve stenosis patients before a valve replacement and after a valve replacement. And we found in this particular study that there were a number of different inflammatory factors that were differentially had differential abundances in serum from post-TAVR and pre-TAVR patients. So we found a number of different factors that seem to control inflammation. So these factors seem to be regulated differently before and after valve replacement. So we were able to characterize the proteome there. And interestingly, culturing cells using the serum from aortic valve stenosis patients can give us these patient-specific myofibroblast phenotypes. So here on the left, so we have these images here where we culture cells from, let's say, pre-TAVR serum and post-TAVR serum from the same patient. And you can immediately appreciate that serum from pre-TAVR serum can activate cells to a myofibroblast relative to serum from a post-TAVR patient that maintains this quiescent phenotype. And this degree of deactivation was patient-specific, right? So we can see a number of different patients that we tested the serum from, and we saw patient-specific myofibroblast activation levels. And why is this data clinically relevant? We were able to show that by culturing cells with patient serum factors, we were able to correlate our in vitro results with in vivo patient outcomes. So we obtained clinical data from our cardiology collaborators and found metrics like aortic valve area and other metrics measured using echocardiography that, for example, in this particular scenario, a reduced aortic valve area indicates more severe stenosis. We were able to correlate that more severe patient with a higher myofibroblast activation level in vitro. So this provides, by culturing cells with patient serum, I think provides a really nice bridge between in vitro disease modeling and in vivo patient outcomes. So given these patient-specific responses to inflammatory factors, our lab next started to think about the question as to whether or not inflammatory factors regulate sex-specific cellular phenotypes during aortic valve stenosis. So like I said, this is our first paper from Brandon's lab. So we have a tradition in our lab where so I make a custom cake for our students with their paper titles and such. But I have the grad students pick a picture for the students to feature on the cake. So it's kind of a fun little tradition. But and then we print like our favorite figure from our paper. But we're super excited that this is like one of our first papers, and we published a couple other articles as well. But this is our first paper from the lab. Exciting. So what did Brandon do to kind of identify clinically relevant inflammatory proteins that might regulate these sex-specific processes? So he took this kind of a computational approach where he first determined correlations using previously published data. So he took our original proteomic data set and took the proteomes, took our serum proteome data and compared them to clinical scores of aortic valve stenosis, so our echocardiography data. And he also took the serum proteome data and compared them to our in vitro activation levels of myofibroblasts. And from this approach, he was able to identify protein candidates that showed correlations in both patient disease severity and myofibroblast activation. And then he was able to validate these candidate proteins using different in vitro experiments that I'll go into details. So just to simplify it again, he basically took our serum proteomic data, compared it to clinical scores of ABS, and generated correlations. Then he took our previously published in vitro data sets and compared them to the serum proteome and identified correlations, and then compared and found overlaps between these two data sets. And he found 38 proteins that are clinically significant proteins that might be drivers of ABS progression and also might be drivers of in vitro myofibroblast activation. So we thought that this was a nice list of proteins that might be regulating, that might serve as a list of biomarkers for aortic valve stenosis, but also elicit changes in the myofibroblast phenotype as well. So one protein that we became very interested in is NXNA2. So we became interested in NXNA2 based on previous work suggesting that this protein is upregulated in calcified derived matrix vesicles, and there's links to calcification in smooth muscle cells. So we thought that this protein, based on the literature, was a nice protein to investigate. And so we looked at the protein abundance of MXNA2, and we compared them to a number of different patient morbidity scores, including the STS score, where a higher STS score indicates a more severe patient. And we found correlations with our MXNA2 data sets. And then with our in vitro myofibroblast activation data set, we also found that increased concentrations in MXNA2 led to increased myofibroblast activation in vitro. We also became very interested in this protein known as cystatin C. So cystatin C is based on, so we became interested in this protein based on previous work suggesting that this protein is linked to increased cardiovascular disease risk and has been shown to contribute to other fibrotic diseases, including the lung. So we also found that abundance measurements in these patients, and we found that abundance measurements of cystatin C in sera correlated to patient measures of disease severity, including and also correlated to myofibroblast activation levels in vitro. So now armed with these kind of hydrogels, armed with our hydrogels and armed with serum, Brandon hypothesized that an XNA2 or cystatin C would drive increased myofibroblast activation in female VIX and osteoblast-like activation in male VIX. So the experiment that he sought out to do was to sex-separate male and female cells and culture them on our soft hydrogels that maintain myofibroblast quiescence, and then dose in either an XNA2 or cystatin C to evaluate how that inflammatory factor changes the myofibroblast phenotype. And he specifically hypothesized, once again, that in female cells, when you dose in these factors, you would observe increased myofibroblast activation. And then when you dose in the factors in male cells, you'll see this increased osteoblast-like differentiation. And the outputs that we used to look at these two phenotypes included looking at stress fiber formation and based on alpha-synuclein gradient mean intensity. We also looked at the osteoblast-like phenotype by quantifying RUNX2 nuclear localization. So RUNX2 is an osteogenic transcription factor that localizes to the nucleus during osteogenic differentiation. So we looked at those two features. So the data revealed that an XNA2 promotes this sex-specific myofibroblast activation and osteoblast-like differentiation phenotype. So we routinely observe that higher doses of annexin-A2 cause increased myofibroblast activation in female VIX. So you can see that data set here. But then, so this is our data on the left are our alpha data, and our data on the right are our RunX2 data. And at baseline, when we dosed in annexin-A2, we did not see a response in the female cells, but we saw an increase in RunX2 nuclear localization in male cells. We also found that higher doses of cystatin C promotes this increased myofibroblast activation in female VIX uniquely. So in our myofibroblast activation data set, you can see that when we dosed in cystatin C, we see this unique increase in myofibroblast activation in female cells relative to male cells that seem to be pretty flatlined. And then in our RunX2 data set, we observed increased RunX2 nuclear localizations in our male data set relative to our female data set that seem to be pretty flatlined. So to try to identify these candidate pathways that might regulate the sex-specific behavior, Brandon next performed a K-means cluster analysis on our serum proteins to basically cluster together proteins based on their correlations with echocardiography data and data collected in the clinic. So he was able to cluster together proteins that had common correlations. And interestingly, this particular cluster, cluster 2, that correlated very strongly with STS scores, we found that annexin-A2 and cystatin C clustered nicely in cluster 2. And then with that, with these individual categories of proteins, we next performed a category pathway analysis to try to converge on pathways that might be relevant to each cluster. And we found in cluster 2, the MAPK signaling pathway was one of the top hit pathways in that particular cluster. So then we next validated the MAPK signaling pathway as a potential driver of the sex-specific myofibroblast activation processes. So we took a known drug known to target p38 MAPK signaling, and we used a previously optimized concentration of this drug, and then we would dose in previously optimized doses of annexin-A2. And we found that in our male data set, we see a unique reduction in myofibroblast. Well, we saw a reduction in myofibroblast activation in our male cells when we treated cells with both annexin-A2 and our drug. But interestingly, in female cells, we saw a unique reduction without annexin-A2 or the p38 MAPK inhibitor. And then we also saw a common effect where p38 MAPK signaling inhibition resulted in a reduction in annexin-A2 mediated myofibroblast activation. But cystatin C was a lot more interesting, in my opinion. We found that when we dosed in cystatin C and the MAPK inhibitor, we found that male cells were basically unresponsive to this inhibitor and to the cystatin C at these doses. But our female data set showed unique reductions in myofibroblast activation when we would treat them with the MAPK inhibitor. And we saw unique increases in myofibroblast activation when we dosed in our cystatin C. So once again, this illustrates how important it is to sex separate your cultures, investigate, and whether or not they have, and most likely your cells will have sex-specific effects in response to different inflammatory factors or different drugs. So that brings me to this proposed mechanism for sexually dimorphic responses to annexin-A2. We think that p38 MAPK signaling partially drives this increased myofibroblast activation in female VIX relative to male VIX. And we think that annexin-A2 uniquely drives the RunX2 nuclear localization in male VIX. And in our cystatin C data sets, we think that cystatin C increases myofibroblast activation uniquely in female VIX through the p38 MAPK signaling pathway, relative to male VIX, where we think that the cells responding to cystatin C drives this osteoblast-like phenotype via an unknown pathway. We still are actively investigating that. So that brings me to kind of just the conclusions for our talk. So male and female myofibroblasts respond differently to hydrogel stiffness and drugs, so please sex separate yourselves for your next experiments. Human sera and hydrogels might serve as a useful tool to generate clinically relevant sex-specific in vitro models. And biomaterials, I think, are a really cool tool to enable precision medicine and sex-specific research. So now I'd also like to talk about how I've been involved a lot in improving the inclusion and diversity of the biomedical workforce and in biomedical engineering. And the title of this slide is called Latinx are underrepresented in engineering. So let me walk you through a little bit. So in 2018, the Engineering by the Numbers report for the American Society of Engineering and Education suggests that only 11.4% of Hispanics obtain bachelor's degrees, and 6.9% or 6.0% of doctoral degrees are awarded to Latinx students. And furthermore, as of 2016, there are only 587 tenured or tenure-track faculty in Latinx engineering departments out of a total of 27,000 engineering faculty across the country. And only 48 of those are Latinx faculty that were born in the United States. So I actually identify as one of these Latinx faculty that was born in the United States. So my family is originally from Colombia, but I was born in Albuquerque. So it's kind of crazy to think that I'm one of these 48 out of the 27,000 professors that identify as US born Latinx. So again, it's a systemic problem where there's underrepresentation of Latinx population in biomedical engineering. So to address this problem, I co-founded this organization known as Latinx Indian in February 2019. So we're a community that leverages digital tools and social media to support Latinx scientists and engineers in the biomedical engineering field at all career stages and try to promote the diversification and inclusion of Latinx scientists in the biomedical workforce. So we have 3 goals. We try to promote and support the professional and personal development of all Latinx and BME. We increase the representation of Latinx at all levels in our profession, and we build a community of peers to feel connected through scholarship and friendship. So, and honestly, this really developed as kind of a collaboration between one of my best friends, Ana Maria Wallace. She's a professor at the University of Florida. So she's the other co-founder of this organization, and it started at a Gordon Research Conference. So that's when I first met Ana. And then at this conference, we met Tatiana Segura and Juan Neco, and we're all Colombian. And we found this Colombian flag randomly at the conference, and we're like, how fun is this? So let's take a picture. And when we all went home, we were just like, oh, it's kind of a bummer. We're all by ourselves now. So we decided to start a Slack group to basically just stay in touch, right? It was just a messaging group. But it ended up morphing into this really beautiful mentoring platform, where now we have over 450 members. And so one of our exec boards, so Valentina Dargam, she's our vice president. And so Ana and I have stepped down from leadership, well, not stepped down, but we're on the advisory board now. We want to pass the torch on to new leadership. So this is our new exec board that are putting on some really fabulous events, for example, navigating grad schools, grad school applications, and applying to faculty positions. We put on a number of different professional events. And out of these different events, we've been able to produce new faculty from our organization. So we have a number of different Latinx Indian members that have pursued faculty positions, which we think is a true testament to the community that we're trying to build and reinforcing support of Latinx scientists in the biomedical engineering community. And again, so I wanted to give a shout out to FIU once again, and to Josh specifically, and your former chair, Ranu, who also helped us so much in organizing our first Latinx and BME, Latinx and BME symposium. And this took place in 2021 virtually, where we had our keynote speaker and a number of different panels. And this is just such a fabulous connection, and I will always hold FIU close to my heart. So you guys were so fantastic in hosting this for us and collaborating. And we hope to host more regular symposia near BMES, right? So we're starting to have conversations about maybe having our 2023 symposium in Seattle. So these are kind of ongoing conversations that we're having with our leadership team. You can read more about our organization. So Ana and I published a perspective piece in Nature Review Materials talking about basically setting up a blueprint for how you can kind of start your own organizations about how to promote inclusivity in the biomedical workforce. And again, I think that we've just really successfully built this beautiful community for all Latinx and biomedical engineering to try to counter feelings of isolation and exclusion and really try to foster community building in our field. So I can safely say that co-founding this organization has been one of the best things I've ever done in my career, because I've gotten to meet so many wonderful people through this organization, and it's just it's definitely the highlight of my professional career. So thank you all and there's a number of different members in this audience as well, so I'm going to give a shout out to everybody here for coming to my presentation. I'd also just love to thank a number of different folks, especially my lab. So we have a lot of fun. So Brandon, Nicole, Talia, Rianne, they're my 4 graduate students, and Mason, Christy, Megan, Rian, and Sarah are my 5 undergrads, so it's been super fun to start the lab. A number of different collaborators at UCSD, and I'd like to thank my funding sources, and I would be more than delighted to take any of your questions. Thank you so much. And thanks for being good sports about not knowing what the titles are.
Wonderful talk and absolutely fascinating science. I'm curious about your future applications and future directions. You've done a lot of tremendous work on the cell cultures. Are you planning to now expand into animal models, preclinical models, use human databases, or maybe sampling to compare it to? Because I'm curious about how do you further go to study the sex differences on a biological level, in the sense, in animals, for example, NIH already requires and frowns on applications that do not actually use both male and female animals, right? And if they, but at the same time, if they do use female mice, for example, they frown on the studies that do not go one step further and to actually track the estrous cycle of them. So what about your future plans? How do you plan to strategically to approach that and to study when is the actual biological effects that you're seeing are more or less relevant? And also, if you're gonna actimize animals, how does that change as well?
It's a good, awesome question, and I'm gonna answer it in 2 ways. So on the animal studies front, I'm collaborating closely with Art Arnold. So Art Arnold, he's a professor at UCLA that has developed this model known as the 4-core genotypes model. And I'm not presenting on this data yet because it's super new and it's super exciting. But the 4-core genotypes model is basically takes a transgenic male mouse where the gene on the Y chromosome has been relocated to an autosomal region on chromosome 3. And you take that transgenic male and cross it with a wild type female, and you can get 4 progeny. You can get male mice with testes, but they have XX or XY chromosomes, and female mice with ovaries that have XX or XY chromosomes. So you can start to understand how X chromosome dosage influences all sorts of phenotypes in the mouse independently of hormones. So that's how we're getting at the mechanisms by which X chromosome dosage is contributing to these phenotypes. But it's not perfect because mice don't translate well to humans, right? So how are we going to do this in humans? I have a collaborator, Jane Ronda Allen, who's collecting blood samples from Turner syndrome patients. So Turner syndrome females have 1X chromosome. So we can begin to understand how 1X chromosome in Turner syndrome patients versus females with 2X chromosomes can potentially have different responses to different environments. So that's our lab's approach on how we're tackling this mechanistic problem that you're suggesting.
Thank you.
Sure. Yes.
Thank you for the talk. That was a lot to digest, but I think we laid it out very, very well.
Thank you.
On the experiment where you did the serum, the patient extracted serum pre and post TAVR, and put it on the cell cultures. Those weren't the patient's cells, right? Those were.
No, that's a great question.
Right. So I guess my question is, that was my first question, one. But then the follow-up would be, were there sex differences there? Like, would you take a female serum and put it on a male fibroblast culture, and were there any changes there?
Yeah, so I didn't touch on those data just because it would have been way too much.
Yeah.
But we have, so to your first question, we use porcine mix, so porcine cells, and we treated them with our human serum. So it's really hard to get good human mix. Like that's like, it's a chronic problem. And my lab is looking at iPS-derived mix, like we're trying to resolve this issue there. But, so we use porcine mix for these models, which is also a testament to porcine mix and being, I think, biologically relevant and could potentially be a useful tool to mimic human disease, right? But to your second question as to whether or not we observe sex differences, of course we did. We cultured male cells with male sera and female cells with female sera and saw that our in vitro models were able to recapitulate sex-specific differences in how cardiac tissue remodels after valve. a paper and happy to answer any questions about that also. Yeah, thanks. Yes?
You use soft and stiff substrates to culture your fix on. And I was wondering why you chose those stiffnesses, right? I think it was 6 fifths on 25.
Yeah, so it's based on AFM data from various valve tissues where this is previous work that has been done to characterize the stiffnesses of valves are very heterogeneous, right? So we're talking like average stiffness of the valve leaflet. So folks have used AFM to quantify, we'll say, an average elastic modulus of these healthy and fibrotic tissues. So we engineered our hydrogels to target one specific stiffness. And our goal was to more or less choose a stiffness that would result in very high activation and very low activation. But I'm sure that if we were to tune the hydrogel and polymer component of our hydrogels, we would get the heterogeneous cell populations that we would be observing in valve tissues in the AFM tissue. Yes?
What would you propose maybe would happen if you changed, like, kind of like on the same regard, if you change the stiffness of the hydrogels, like, do you think it would give you maybe different results if it were more stiff versus if it were less stiff, or if you were to use maybe a different medium in the hydrogels?
So yes, so we can certainly tune the hydrogel stiffness to observe sex-specific effects. And we're actually working in this area right now where we have hydrogels where we can do dynamic stiffening. So we can culture a cell on a soft hydrogel and shine UV on our gel to cure it and stiffen it. And we're observing sex-specific cellular behaviors on how these cells respond to dynamic stiffening processes in vitro. So yeah, to your question, we can tune the stiffnesses in real time and start to investigate how the cells respond to matrix stiffening in a dynamic way and what those sex-specific pathways might be. Thank you.
This question might be very abstract, but you were talking about the nice, and so on your progeny. But with progeny, it's almost like probability-based reproduction. So is there a way that you kind of account for that, or do you just like, is there just a whole bunch of?
I'm sorry, did you say AT-based reduction?
No.
I'm sorry, I didn't hear you.
Probability-based reproduction, like it's based on
Reproduction, yeah. Okay, sorry, yeah, I misunderstood you. So the progeny, we can get into more details of this, but the XX males and the XY females are sterile. So their ability to reproduce are eliminated through this cross. But I would suspect that to potentially influence our phenotypes, but we can certainly talk about that later. Yes?
Impressive work.
Thank you.
It made me a little anxious.
In what way?
Well, if we take the premise seriously that sex differences really matter in therapy, and by extension, lifestyle, immunology, hormones, and perhaps their interactions. So it creates this large matrix where every cell potentially calls out for its own specific treatment variant, and it takes the career of an impressive person to isolate the pathway in one of those cells. It's difficult to understand how that scales. Is that something you worry about?
Oh, absolutely. So, I mean, you're getting at the question of patient-specific heterogeneity, right? But you're getting at a cellular heterogeneity, right? So there's heterogeneity in all scales, right? But as engineers, our responsibility is to figure out what variables are most important to be looking at, right? So I don't think that I'm envisioning a world where we treat every cell or we treat every patient uniquely. That's just not possible, right? There's 8 billion people in the world. Like, we can't generate 8 billion therapies, right? It's kind of like what you're suggesting. But I think what we can do is start to logically think about what diseases can be targeted with generic treatments, but also recognize that there are some diseases that are sex-specific. So there might be a simple tweak that we can do to treatment protocols, like maybe a combo drug for females instead of a like one drug for males, right? That's the impact that I'm trying to go for, and not so. And thinking about like, and that would be tremendously impactful, right? Because we're targeting, we can treat half of the population more effectively. But I'm not trying to suggest that we should go the precision medicine route specifically, because it just becomes impractical at that point, and like not very precision. It's like it's do I think males have? That's a good question. So I think that I can draw analogies to, for example, women experiencing osteoporosis. So there's this one protein known as osteopontin. Osteopontin is a protein that and osteopontin regulates calcium production, right? So when osteopontin essentially depletes calcium, right? So when that's expressed more highly in females, we think that that could be a potential mechanism by which they're reducing calcium overall in women, especially in menopause, right? But that's not the only part of the story, and I think about this a lot. I'm talking about this more because women eventually do experience calcification of ducts. Like it's not like they don't just experience fibrosis. Like they have this. I kind of think of it as a latent period where they're like fibrotic, and then they calcify. Whereas men just pretty much calcify right away, right? So there's got to be a hormonal aspect to it as well. But this is definitely a complex problem that I think is definitely an area.
Ask one. Okay, so
Yes.
I mentioned the mechanical sensitivity difference, right? So a couple of questions there. So one, is there no difference in stiffness between male and female valves just at baseline? And then the second part is, what about deformation? So not just stiffness, but the fact that you have dynamic tissues. What does that mean?
We quantified valve stiffness with using shear rheology, probably not the best technique, but at baseline. And we didn't see any specific difference as far as, or at least like technical differences in the mechanics of the machine. Other folks, including Danielle also looked at not just valve tissues, but cardiac tissues as well. She's been able to biaxial stretching of these tissues, and she has their speculative effects there. My lab is also looking at how collagen is organized within the tissues. And even though there isn't a sex difference in stiffness, we do see a sex difference in collagen orientation of structures. Because I think that might be more relevant to how a cell is experiencing the initial matrix in these tissues, and then when they become more diseased and those sex differences can be exacerbated, it's stiffness.
We should talk more about that.
Yeah.
Yeah.
And then within the, so on the deformation front, so I mean, because everything you're looking at right now is static, right? Which is certainly important in terms of difference in mechanical sensitivity, but
Right.
you're seeing fairly large strains in these tissues that also play a role.
I also think that the tissue, the stiffnesses can be exacerbated with inflammatory factors. So we have some early data suggesting that when we dose in inflammatory factors like beta, we see uniquely that female tissues contract crazy relative to male tissues. So I would anticipate that tissue is very different, right? So there's lots of things to think about there.
Any other final questions? If not, let's thank Dr. Aguado again.
Thank you.